Welcome, everyone, and thank you for joining us today. My name is Rohit Mehra, and I'm honored to open this GUPS Distinguished Lecture Series event session organized by our GUPS Education Committee. At GUPS, our mission is rooted in a commitment to improving patient care through education, research, and the promotion of best practices in urologic pathology. We are proud to serve as a hub for collaboration and innovation among professionals dedicated to genitourinary diseases. So today's event is special, and this is part of our new distinguished lecture series, a cornerstone of GUPS programming. Through distinguished lecture series, we learn from visionary leaders in the field like translational scientists, clinicians, and pathologists whose groundbreaking work continues to shape our understanding of genitourinary pathology. These leaders are not only prolific contributors to literature, but also active voices in shaping the future of cancer research, clinical care, and pathology. Their insights have indeed redefined how we approach tumor biology and diagnostics. Today's session, in particular, addresses a timely and complex topic of urothelial carcinoma pathogenesis, insights from genomic analysis, and whole organ mapping. This session is being moderated by my outstanding GU colleague and GUPS Education Committee member, Dr. Hikmat Al Ahmadi from MSKCC. I'm now requesting Dr. Al Ahmadi please welcome and introduce our distinguished speaker, Dr. Bogdan Cherniak, who has kindly agreed to bring his expertise and discuss this topic with us. Over to you, Hikmat.
Thank you, Rohit, and welcome everyone. Welcome you again for this inaugural Distinguished Lecture series. I couldn't be more thrilled to have Dr. Cherniak be our first speaker for this event. Before I introduce Dr. Cherniak with a few words, I would like to remind you and encourage you to submit your questions within the Zoom Q&A tab throughout the session, and we'll get to them towards the end, and our distinguished speaker will be addressing them one by one as we get to the end of his talk. So for Dr. Cherniak, he's one of the few in the field with a status that doesn't really need introduction, as he's a real thought leader and prominent presence in bladder cancer research throughout his career. But I'll say a few words just to get familiar with him as a person, as a scientist, before I turn it over to him. So Dr. Cherniak is currently a professor of pathology at the MD Anderson Cancer Center and hold the Nathan W. Listeter Distinguished Chair in Urology. He hails from Poland. He's a proud Polish American, where he was born, and before coming to the US to continue his career. Throughout his career, he has distinguished himself in both diagnostic pathology and a multi-institutional based research related to both genitourinary and sarcoma pathology. Throughout his career, he received multiple NCA and NIH R01 and U01 grants given to study the molecular biology of bladder cancer. Has been honored with several prestigious institutional, national, and international awards, including the Southern Medical Association Moreton Award for Original Research and the 2012 Nathan Kaufman Award and Lectureship from the United States and Canadian Academy of Pathology. He's authored over 300 peer-reviewed articles, many chapters in books and textbooks, as well as many review articles, and many of these papers are focused on the biology of bladder cancer and were published in really high-impact journals, including Nature, Cell, Cancer Cell, Cell Reports, the GNCI, and eBiomedicine. He was one of these contributors and co-author of the two major papers that came from the Cancer Genome Atlas analysis working group and bladder cancer that were both published in Nature and in Cell. But perhaps most significantly, or the most important contribution of Dr. Cherniak, is his approach to bladder cancer, where he did a unique a study that took a while, which is focusing on whole organ mapping, both histologic and genomic, comprehensive analysis of bladder specimens to study the events of early bladder cancer development. Throughout this work, he discover new class of forerunner genes whose inactivation is required for the subsequent silencing of the major tumor suppressor genes like RB1 and B53. And this sequential combination of events initiates cancer development. This discovery represents a paradigm shift for understanding cancer development and an entirely new field to identify molecular mechanisms of early human carcinogenesis. So without further ado, turn it over to you, Dr. Shonyak. Please go ahead.
I would like to thank the Genitourinary Society for the invitation. It is a great pleasure for me to talk to you. And as it was mentioned before, the subject of my presentation today is the pathogenesis of urothelial carcinoma. And we will address it on the whole organ scale. And the center of this presentation and the unifying theme is the novel whole organ mapping approach and what kind of new and unique information concerning bladder carcinogenesis we can get from this complex analytical approach. This lecture will have three major parts. At the beginning, we will discuss briefly dual track concept of bladder cancer development. It develops through two distinguished tracks. Then the mid portion of my presentation will focus on modeling of bladder cancer evolution from mucosal field effects on the whole organ scale. And the last part will focus on the concept of forerunner genes, which were also identified by the whole organ mapping and unique analysis of sequence events around the RB gene region. So let's start with the dual track concept of bladder cancer development. This is credited to my mentor and a person who attracted me to study bladder carcinogenesis, Dr. Leopold G. Koss. He performed in 60s and early 70s the first prototypic whole organ mapping and identified that bladder cancer develops through two distinguished tracks or pathways, papillary and non-papillary. The most common form of bladder cancer is the papillary superficial lesion. And if we take randomly selected, let's say 100 samples of bladder cancers, nearly 80% will be low-grade superficial papillary lesions. A smaller fraction, around 20% of them, 15 to 20%, are high-grade non-papillary lesions. This is the highly aggressive group of the disease. And this two forms of bladder cancer develop from distinguished or distinctive field effects. Papillary tumors developed are usually molecularly luminal, and they develop from the unique field effect defined as luminal field effect. The precursor of the high-grade invasive bladder cancer is a carcinoma in situ, and it develops from the so-called basal field effect. A small fraction of superficial papillary tumors, around 15 to 20%, typically after many recurrences, will eventually progress to high-grade invasive disease. Typically, this group of patients with high-grade invasive cancer do not have a history of prior papillary lesions. Okay? This progression from papillary tumors is a relatively rare event, and it happens after many years of recurrences. These two groups of tumors require completely different approaches. The superficial papillary, typically low-grade tumors, require lifetime surveillance. Okay, the high-grade invasive carcinomas require completely different protocol in which extensive surgery, cystectomy is performed combined with other treatment modalities. So let's look at examples of the precursor lesions. At the bottom, we have a morphology of normal urothelium, and in the middle portion is low-grade intraurothelial neoplasia. clonar hyperplastic conditions in which the urothelium is still organized. The nuclei are a little bit larger, a little bit darker, but they have overall preserved organization, and at the surface we see terminally differentiated urothelial cells. And this type of lesion is a precursor of low-grade superficial papillary tumors. Another in situ precursor collision defined as high-grade urothelial neoplasia, commonly referred by pathologists as carcinoma in situ, is a precursor of high-grade urothelial carcinoma with high propensity for invasive growth and distant metastasis. Look at the high degree of atypia, disorganization of the urothelium, and multiple mitotic figures. This is an example of the conventional low-grade superficial urothelial carcinoma. The lesion, which is the precursor of this condition, is practically speaking microscopically identical to the urothelium that is covering the papillary structures. An example of high-grade invasive non-papillary carcinoma, this form of the bladder cancer has high degree of microscopic diversity, frequent glandular differentiation, squamous features, and practically speaking, more than 20 distinctive microscopic types of the disease have been described in the literature. So when we look at the molecular level, there are several systems of classifications. Okay? Today, I will attract you to the MD Anderson base, the one that we invented, which separates the bladder tumors into two major groups referred to as luminal and basal. The concept of this classification is based on the pattern of expression of markers in normal urothelium. When we look at the diagrammatic drawing of the urothelial, of the multi-layered urothelium, we can divide this into two major layers: the undifferentiated basal cells and intermediate plus superficial cells, which have various degrees of urothelial differentiation. So tumors with microscopic features corresponding to intermediate and terminally differentiated cells are called luminal, and those which reflect a phenotype of the basal undifferentiated cells are called basal. We have over 100 markers which are distinctively expressed in normal urothelium. Here are signature exemplary markers. The signature basal markers are keratin, the so-called basal keratin, keratin 5-6, okay? On the opposite side of the spectrum, the markers of terminal urothelial differentiation are uroplakins, okay? And there are many other markers. By selecting a system of luminal and basal markers, we can divide bladder cancers into luminal and basal categories. There is a small fraction of tumors which are losing both groups of markers, luminal and basal. They are called double negative. They represent a small fraction of poorly differentiated basal carcinomas as they retain the expression of the signature basal marker CD44. We also have an algorithm of quantitative assessment of luminal and basal differentiation. It is called basal to luminal transition score. You can see that the vast majority of luminal tumors have the positive BLT score, and the basal tumors have a negative BLT score. So let's move on to modeling of bladder cancer evolution from mucosal field effects on the whole organ scale. Why do we need this kind of complicated and laborious approach? Let's look at this. Okay? This is a Hullman specimen. It is a historical specimen of human bladder prepared by Dr. Schade from Great Britain in late 60s. Okay? It shows multifocal papillary tumors. All of them are superficial. This is the urethral orifice. If we look at them molecularly, it is clear that all of them are clonar. So where is the original clone? An old concept of antiquity was that the largest tumor was the primary and the smaller one were implants. Okay? This is historical concept. We do not believe that these multifocal lesions develop like that. If you look carefully, you will notice that they are multiple. The entire bladder mucosa is studded with multiple small microscopic papillary lesions. So the original clone is here in the field effect of the entire bladder mucosa. So if you want to ask the question, what initiates bladder carcinogenesis, the answer for this question is in the field effect, in the bladder mucosa adjacent, not in the tumor. So this is how we prepare whole organ maps. We develop a specific mapping grid, which is pressed against the open cystectomy specimen. This is a large tumor on the posterior wall. And this mapping grid has a unique construction. It has a groove at the bottom. So these are the wells, usually 1 by 2 centimeters, from which we extract DNA, RNA, and sometimes we can complement this with protein and metabolites. So we extract and prepare urothelial cell suspensions, and we end up with between 30 to 40 geographically oriented samples, which are specifically anatomically oriented to the bladder surface. Okay? When this is completed, this grid is lifted, and it leaves permanent impression, and the bladder is fixed overnight, and then morphology is reconstructed from these grooves, which preserves the microscopic features of covering urothelium and tumor for microscopic examination. These urothelial cell suspensions are prepared by filtration and trypsinization, and we end up with clear, almost tissue culture quality urothelial cell suspensions, somewhere between 30 to 40 per one bladder. And these are the examples of the normal urothelium, low-grade dysplasia, carcinoma in situ, invasive cancer from the same case. Even very experienced pathologists will not notice that we've done anything unique. The morphology of the urothelium and invasive cancer is so well preserved. So now what is the advantage of this approach? This approach provides us with the geographic distribution of individual molecular events, and we can analyze this on the whole genome scale, and which are superimposed on the histologic map of the bladder mucosa. These dark areas correspond to urothelial carcinoma, and these lightest areas are microscopically normal urothelium. Everything in between represents either low-grade intraurothelial neoplasia or high-grade intraurothelial neoplasia, or simply carcinoma in situ. So let's look at this concept of luminal and basal molecular forms of bladder cancer. We have approximately in our collections 27 samples, and our maps are numbered. So this is map 19, and this is map 24. And I will label them, and on the left side is luminal, and on the right side is basal. So we have two groups of markers, the same as one as shown on the TCGA cohort a couple of slides before, luminal, basal, and we have representative target genes of the GATA3 and PPAR gamma pathways, which are signature transcriptional factors involved in luminal differentiation. And we have representative target genes of P63, which is the representative or signature basal marker. Look at the color. Luminar markers are red, and these are individual clusters. There's 40 samples of the bladder mucosa, and this dark and yellow are carcinoma in situ and bladder cancer, which cluster together. So all luminar markers are red. This is the most reddish, most highly overexpressed marker, which is in this part, in the instance keratin number 20. GATA3 and transcription and PPAR gamma target genes are overexpressed, and then basal markers and basal transcription factors are downregulated. So what we can say that luminal form of the bladder cancer develops from the luminal field effect. And the opposite is in the example of basal form of bladder cancer. Cancer samples are clustered here. This time, all luminal markers and luminal transcription factors are downregulated, and we have an upregulation of basal markers. The strongest upregulated markers is one of the basal keratins, keratin 6, and here is the upregulation of P63 target genes, signature basal transcription factors. So basal cancer develops from the basal field effects. And here we have a quantitative assessment by the basal to luminal score. And here in the field effects, all scores are positive, suggesting or consistent with luminal differentiation. And here are negative. 0 is here, consistent with basal or lack of luminal differentiation. So we have this data on an RNA whole genome transcription scale, and using this mapping on the entire bladder mucosa, we can actually analyze them in a variety of ways. Here we have an example on single chromosome, genes mapping to these chromosomes, and they are displayed in the form of three-dimensional diagrams forming plaques. And the whole organ map of the bladder mucosa with dark areas corresponding to cancer is shown here. And the overexpressed genes are forming red plaques, and the downregulated genes are forming blue plaques, okay? So when we analyze this on a total genome scale, we can actually identify the sequence of events. These are the top upregulated and downregulated genes. Blue is downregulated, red is upregulated. In the initial field effects, we use the word monotonicity. Monotonic in our case means that they are upregulated already in areas corresponding to normal bladder mucosa, and they continue to be upregulated with progression to carcinoma in situ in invasive cancer. And using this kind of data, we can analyze the sequence of events in progression of the disease through paraneoplastic in situ conditions to carcinoma in situ and invasive disease. Now, what is going to happen if we sequence DNA on the entire organ scale? We have another example, 40 samples divided into normal urothelium combined with low-grade intrauterine neoplasia, carcinoma in situ, and urothelial carcinoma. We can identify in a single bladder between 1,000 plus to over 40,000 mutations. So the amount of data, this generates terabytes of data. So by clustering and analyzing the frequency of the mutant DNA, we can separate the mutations into two major clusters. One group corresponds to mutations which are restricted to individual mucosal samples. We call them alpha mutations. And then here is the group of mutations which are spreading across almost entire bladder mucosa. They are separated here, and we can see two distinctive groups of these mutations. One in which mutated DNA is typically below 10% of the total DNA. And these are beta mutations. And then finally those which are expanding clonally, and red corresponds to something that is more than 40%, okay? And the cutoff, but some of them represent nearly 100% of mutant DNA. You are used to see this kind of diagrams with 3 or 2 points. This is the phylogenetic tree generated from this particular case. And typically, it's a primary tumor, 1 or 2 metastases, so this is some kind of V-shaped diagram. Here, we have around 40 samples, and this universally generates complex branching phylogenetic tree. Each of these nodes represents a new wave of mutations. And Here we have three major branches, delta, epsilon, and zeta. And when we look at this alpha, beta, and gamma mutations, they are not evenly distributed in these three branches. In this particular branch, in branch zeta, contains the majority of gamma mutations. So something unique is going on. So now let's look at how these mutations, alpha, beta, and gamma, are distributed over the bladder mucosa. That's where the whole organ mapping comes into play. So alpha mutations are restricted to individual mucosal samples. Beta mutations are regional. They expand and involve some areas of bladder mucosas. And then finally, gamma mutations are spreading across the entire bladder mucosa. They form plaques involving just missing few samples. If we look at the content of the so-called cosmic mutations, these are the mutations which are believed to be oncogenic. There is a dramatic increase of cosmic mutations when we progress from alpha through beta to gamma mutations. Around 50% of mutations in gamma category are actually cosmic mutations. Now, here the complexity starts. This unique distribution of mutations permits the so-called time modeling, which is otherwise not possible. So this is the fundamental equation which is used to assess the time of the mutations. And this extensive computational analysis are done on each mutation, many thousands and many samples, okay? This is the computation of terabytes of data. And the age of mutations forms a peculiar curve, this kind of inverse slope. And I would like you to look at this number. So these mutations developed over 30 years. So the mutational signature is not 1, 2, or 3 years old. It is, we are working on this during our lifetime. Okay? Now here are all mutations put on the time modeling scale. Here we have alpha, beta, and gamma mutations separated. It turned out that alpha mutations are the oldest, and they develop gradually through 3 decades before the diagnosis of cancers. Beta mutations are much younger. They are 5-year-old approximately. And then the youngest are the gamma mutations. We believe that these are ultimately drivers of the progression to invasive cancer. So when we look at the diagram of all mutations, we can put hypothetical errors where these 3 classes of mutations start. Alpha are the oldest. This turning point is the advent of beta mutations. And using this point, we can divide bladder carcinogenesis into dormant phase and progressive phase. And finally, the gamma mutations, 2 to 3 years old, which are ultimate progressors, ultimate drivers of the disease to the invasive cancer. So the problem started at this moment. Okay? We have a specific program in which we are trying to identify these 3 classes of mutations in voided urine. If you like my talk today, I will be open to give you another talk when we will be talking about the practical implications, how actually this approach help us to identify early detection and early treatment markers for the disease. So if we can identify in a non-invasive way this turning point of changing from dormant phase to progressive phase, we can change the fate of the patient. Now, move to the copy number variations. Things are getting complicated, and they are not going to get easier during the progress of this presentation. An example of one chromosome, chromosome 8, and we have copy number variations analysis in all 40 mucosal samples. Red is the amplifications, and you can tell that the long arm of the chromosome 8 is amplified when we progress to carcinoma in situ. And at the same time, This tumor is losing the short arm of the chromosome. This is the three-dimensional model of copy number variations of the entire chromosome, and this part is enlarged here. I would like to attract your attention to these two areas indicated by dark arrows. This is happening with progression to carcinoma in situ, but there are smaller areas which are lost already in areas corresponding to microscopically normal urothelium. And this part of the chromosome is enlarged here. These are these two spots which are being lost already in the entire bladder mucosa, and these are representative genes which can be identified. These are our potential markers for early detection. So now just envision that this is the type of data that we are getting on a single chromosome from a single whole organ map. What kind of diagram we can generate if we put this on a total genomic scale? Here it is. Okay? This is the copy number variation model of bladder cancer using single cell cystectomy specimen. These three belts of samples correspond to normal urothelium, low-grade intraurothelial neoplasia, carcinoma in situ, and urothelia and invasive carcinoma. Here is our chromosome 8 from the same case, loss with progression to carcinoma in situ of the short arm and gain of the long arm. Okay? And we have changes like that on the total genomic scale. I can probably continue talking for an hour or two just analyzing this single diagram, but we're not going to do this. Let's move forward, okay? So with this kind of data, there are algorithms available. Some of them are actually developed by my statistical partners, in which we can put all the data together and start analyzing the pathways. So this is the combination of multi-pathway analysis, and we can identify by this approach over 100 dysregulated pathways, and its focuses on the early events, those monotonically dysregulated pathways. Here is the summary of the top dysregulated pathways, which can be divided into three groups: immunity and inflammation, signal transduction with the focus of differentiation, and oncogenic pathways. Multiple oncogenic pathways are already activated in mucosal field effects in which microscopically urothelium looks normal. I would like to attract your attention to the p53 signaling. It is already dysregulated in microscopically normal urothelium. Here are the group of samples or group of pathways which are dysregulated, and they participate in urothelial differentiation. You don't know the name of this gene, but on the top of this pathway is the LPAR6 receptor. This is one of our signature forerunner genes, and we will talk about this within a minute or two. So this is when we put together genomic pathways, DNA sequencing, RNA methylation, and mutations. Now, if we add proteomic and metabolomic data to this, a different picture emerges. These are the top dysregulated pathways in which proteomic and metabolomic data on the whole organ scale are involved in the analysis. Let's look at this tallest, most significant bars. They correspond to ATP synthesis, oxidative phosphorylation, and mitochondrial dysfunction. All these three are overlapping. And here is the dysregulation of the energy metabolism exemplified by oxidative phosphorylation. Over 60 enzymes encoded by a number of genes forming 5 oxidative phosphorylation complexes. This is the whole organ map, and microscopically normal urothelium is here, and here is the invasive carcinoma. You can see a massive downregulation of, practically speaking, all oxidative phosphorylation complexes. This is the continuation of this analysis. This is complemented by this regulation of the glycolysis, citric acid metabolism. These are citric acid enzymes, and these are citric acid metabolites. Look at the top. This is the lactate, okay? And lactic acid is upregulated already in microscopically normal urothelium, and a lactate dehydrogenase, the key enzyme that regulates the production of lactic acid. Look at this pattern. These are the outlined areas of downregulation of oxidative phosphorylation enzyme from the mitochondria, which form the plaque. What is the message of this complex analysis? That the famous switch to anaerobic glycolysis, the so-called Farburg phenotype, is actually an early field effect. And this kind of data indicate that this dysregulation of mitochondrial energy metabolism with the glycolysis, the anaerobic glycolysis shift represent probably a major driving force of progression of bladder cancer from field effects to invasive disease. So this concludes the section of the presentation on genomic analysis on the whole organ scale. So let's move to the concept of forerunner genes. Okay? They were also identified by the whole organ mapping. This is the summary diagram from PNAS, and it was published in 2007, long ago. This was mapping of this was done by manual analysis, manual sequencing of single nucleotide polymorphic sites. So this approach, performed on several maps, okay, identified that there is a loss of approximately 5 megabase segment around the RB genes. This loss is outlined here on one example, and it's outlined here. In the vast majority of cases, when we started sequencing RB gene, the RB gene in this map is not mutated. It's a wild type. So this clearly indicates that this clonal expansion, forming a large plaque in the bladder mucosa, was not driven by the RB gene. Something else mapping to this area should be responsible for this. There were examples in which RB gene was mutated. By this mutation, was always restricted to carcinoma in situ, which is here, and invasive cancer. So again, this plaque covering the majority of bladder mucosa is not driven by the mutant. And this is how we generated the concept of forerunner genes. They are named forerunner because they run the field, they promote clonal expansion before the inactivation of major tumor suppressors such as RB1. So in order to identify the candidates, we design a specific microarray, custom designed microarray. It is spanning the entire 20 megabase area, including this 5 megabase minimally deleted regions. And I would like to attract your attention to this diagram. This is the RB gene with axonal structure. And this region is mapped expression pattern with 12,000 probes. So there are several million probes across this region. And this permits the identification of the expression pattern on individual exons. There is something that is downregulated here, and this is our LPAR6 gene, which is located inside of the RB gene, and this gene is here. So if we analyze quantitatively the data, it turned out that the two genes mapping to this region are most severely downregulated. One of them is called LPAR6, and another one is CAP39. So this is the map of the minimally deleted regions with potential candidates, and we have 5 potential candidates. Some others were also downregulated. By the most significantly downregulated is LPAR6. The full name of this gene, lipophosphatidic acid receptor number 6, and CUP39, calcium binding protein 39-like. And we will focus on these two genes. From 2007, when this original mapping paper was published, it took us about 15 years to figure out what is going on in this area and how these genes contribute to bladder carcinogenesis. Let's look at this. These are mouse embryonic fibroblasts from LPAR6 and CAP39 animal knockout models, germline models. All cells in these animals have lost LPAR6 or CAP39. These are mice embryonic fibroblasts, and we are looking at the expression pattern of luminal and basal markers in our mouse embryonic fibroblast. Look at this. Here is redness. So this is LPAR6KO animal. The LPAR6 gene is missing, and we are causing upregulation of luminal markers, PPAR gamma, and downregulation of basal markers. Here is gene set enrichment analysis showing a upregulation of PPAR gamma target genes. The opposite situation in CAP39KO animals. The upregulation is of basal markers, and we are getting upregulation of P63 signature transcription factors of basal cells. This provides preliminary data that these genes are capable of dysregulating target genes which are involved in basal and luminal differentiation. Similar to human tumor cells, bladder cancer cell lines can be divided into luminal, basal, and double negative, except that double negatives represent larger proportion of basal cell lines. Interestingly, a simple test to figure it out whether the cell line is luminal in basal is not DNA sequencing. It's letting the cells to produce urospheres. These urospheres or the spheres in general in epithelial cells are used in stem cell research. It is believed that each urosphere is developing from a singular stem cells, but they also reflect the potential of urothelial differentiation. We grow the cells in adhesion-free medium, and this is an exemplary example of luminal cells, which forms beautiful urospheres. This is electron microscopy. These cells on the surface of the urosphere have some ultrastructural features of umbrella cells. And this urospheres forming cells excrete GATA3, and this is a signature basal cell line, UC6. It's not forming spheres. It grows in the dispersed pattern. It forms loose, discoherent clusters. This is electron microscopy of these clusters. So we ask the fundamental question, since these cells dysregulate luminal and basal genes, which are involved in luminal and basal differentiation, are those genes capable of switching the phenotype or cause the phenotypic plasticity? So we have a signature luminal, or basal UC6 bladder cancer cell line. We developed CRISPR knockout, and look what is happening. This cell, when we knock down LPAR6, is producing urospheres. Beautiful scanning electron microscopy, transmission electron microscopy, and immunohistochemically showing upregulation of GATA3. Here on the genomic level is upregulation of PPAR gamma, okay, and gene set enrichment of PPAR gamma. And I would like to attract your attention that the top dysregulated pathways in the switch involve cholesterol synthesis. In reality, the cholesterol synthesis is downregulated. However, the pathway which is upregulated with this switch, with this phenotypic switch, is upfolded protein response. many of the unfolded protein response proteins are actually heat shot proteins. Right? But this unfolded protein response plays a major regulatory role in epithelial cell differentiation. So look at the opposite side. We are knocking down CAP39L in the luminal cells, and we are switching it to the basal phenotype. And again, original urospheres, and then dispersed growth, which is negative for GATA3, and here is the positive staining for GATA3. And this is associated with the upregulation of P63, and this is gene set enrichment analysis. And this time is the opposite, reciprocal. Unfolded protein response is going down, and cholesterol synthesis is going up. So this was in vitro, in bladder cancer cell line. What is going on in vivo? We have animals with KO of LPAR6 and CAP39. This is single cell sequencing of the urothelium in control wild type animals, animals with KO of LPAR6 and animals with KO of CAP39. Let's look at the electron microscopy. This is normal urothelium. This is LPAR6 KO urothelium. It's hyperplasia, and we see gradual progression from basal to terminally differentiated cells. In CAP39L KO, there is something different, with proliferation of undifferentiated basal cells. This is immunofluorescence, and here are cytokeratin 14 basal keratin proliferating cells. Look at the single cell sequencing data. Using the gene expression pattern, we can separate the urothelial cells into multiple clusters, approximately 20 clusters, and then inflammatory cells and stromal cells are filtered out. And using the luminal basal transcription factors, we can separate them into basal, intermediate, and luminal cells. Look at the proportion of luminal cells in normal urothelium, and look what is happening when we perform this analysis of LPAR6 KO cells. This is an expansion of luminal cells, and the opposite effect, basal cells are expanding in CAP39 KO. So our genes are changing the urothelial differentiation program in vivo. Here they are promoting luminal differentiation when the LPAR6 is missing, and here we are expanding undifferentiated basal urothelial cells when CAP39 is missing. So what is going on when we look at the expression of our LPAR6 and CAP39 genes in normal urothelium? This is human urothelium single cell sequencing. Look, these are basal cells, and CAP39 is missing. And LPAR6 is strongly expressed in these cells, and the opposite effect in terminal cell. If we look at the quantitations of LPAR6 and CAP39, it looks like LPAR6 is expressed predominantly in basal cells and intermediate, and goes down in luminal, and the opposite effect. This is an expression of CAP39 and LPAR6 in individual urothelial cells, clustered according to the differentiation pattern. Here is the area of basal cells, and here are the terminally differentiated superficial urothelial cells. Look at CAP39 expression, predominantly in superficial cells, and LPAR6. This expression pattern is consistent with the major regulatory role of LPAR6 and CAP39 in human urothelium. So the next question is, what is going to happen if we expose this kind of dysregulation of urothelial change to the carcinogen? BBN induction of bladder cancer in animal model with LPAR6 loss and CAP39 loss. A simple story is emerging. LPAR6 KO animals develop myriads of superficial papillary tumors. Some of them are developing invasion, and the BLT scores are positive. These are predominantly luminal tumor. And the opposite thing, this is carcinoma in situ with expansion of basal cells positive for keratin 5-6, and poorly differentiated, almost sarcomatoid carcinomas of the bladder with negative BLT score, which are basal. So this dysregulation of LPAR6 is actually priming the field effect to distinctive molecular forms of carcinogenesis, luminal and basal. So how to summarize this? These two genes play a major role in urothelial differentiation. When LPAR6 is downregulated, cholesterol synthesis goes out, unfolded protein is activated, and we promote luminal differentiation. This kind of field effect sensitizes urothelium to luminal carcinogenesis. The opposite effect, CAP39 goes down, cholesterol synthesis is activated, unfolded protein is suspended, and the basal cells are proliferating. And this sensitizes the urothelium to the so-called basal form of carcinogenesis. These are plaques of hypermethylated CAP39 and LPAR6 in bladder mucosa. So it's time to end everything. And we are having dual track concept of bladder carcinogenesis developed by my mentor, Leo Kost, with the superimposition of novel molecular data. We focused today on the incipient phases of bladder carcinogenesis. One more time, luminal papillary cancers develop from luminal field effects, and basal non-papillary cancers develop basal effect, and the 400 genes actually dysregulate early urothelial in urothelium and sensitizes, directs this carcinogenesis to distinct luminal and basal form. Finally, take-home messages: the development of bladder cancer with luminal papillary and basal papillary tracts, affected genesis, mutational evolution takes nearly 30, and we have alpha, beta, and gamma mutations, and the carcinogenesis based on the age of the mutational landscape can be divided into dormant and progressive phase. Complex alterations of urothelial immunity, differentiation programs implemented with mitochondrial dysregulation initiated bladder cancer. Forerunner LPAR6 and CAP39 contributed to bladder cancer initiation by urothelial differentiation. Thank you very much. That's the end of my presentation.
Thank you very much, Dr. Choniak. That was wonderful talk, tour de force, and bladder cancer development, and just shows how much dedication and effort one has to put through to come up, to be able to discover this type of unique work. So I could see some questions here. So clearly the audience have seen it very, very interesting. And I'll start with a couple of questions. Start with the question from Rahul Manand. He's asking, great insights on time-based modeling of bladder cancer. As an EU question, I'm wondering, in context to clinical samples, which are often snapshots in time, how does this affect our ability to map the slow dormant phase and more progressive phase of tumor evolution? And can we predict somehow this in patient care to capture the temporal dynamics of bladder cancer evolution?
Okay, if I could answer this question, this may take me straight to start. But that's a great vision for actually the entire intention of this approach is to identify the earliest changes of that.
I think we're losing some of your voice, Dr. Choniak.
I will repeat it.
Yeah,
so the
thank you.
concept of this research is to identify the earliest possible phases of bladder carcinogenesis and use them as a potential therapeutic preventive target and for early detection. I will switch to another lecture in order to address this question, because we have a specific program in which we try to identify the age of mutations by the analysis of voided urine samples. The hope is that we'll be able to identify in which carcinogenesis is switching from dormant to progressive phases. You have to give me another year or two, and perhaps I will come back to answer your question much more specifically.
Thank you. So we have another question from Santosh Menon who's asking, would urinary detection of LPAR or CAP39 someday aid in early detection of bladder cancer? So if you can just re-emphasize, Dr. Shiniak, these are not necessarily mutated but hypermethylated. Did I get this right from your presentation? They're not like truncating mutations, it's like silencing through hypermethylation, right? And would that be detected in urine at some point, or do you have a vision for that?
Yes, but now let me give you this. I can answer this question by showing you something like that. These are the TCG cohort. Okay? And here we have a target genes which are upregulated. These are identified in mouse fibroblasts. So look what is happening. So the upregulated genes are upregulated in luminal cancer, and downregulated genes are downregulated in luminal cancer. The opposite effect is in CAP39L. Basal upregulated genes are upregulated in basal. So in summary, luminal tumors are having LPAR6 depleted phenotype, and basal tumors in the TCG cohort have a CAP39 depleted phenotype. This tells you that these are common events. In terms of the methylation of forerunner genes, LPAR6 and CAP39 happens in 30 to 40% of bladder cancers of respective luminal basal type. So I do not believe that using the methylation as effective biomarkers for detection of. That's the end of my answer for this.
Okay. All right, so we have another question. What about the stroma-rich molecular classification of urothelial carcinoma? Can that fit into this model that you have presented to us?
Stroma-rich is basically a contaminant.
I think you're breaking up, Dr. Shiniak.
We are deviating to the consensus classification, and this is basically a combination of tumor cells by stromal elements. And I do not believe that this is a real molecular subtype of cancer. And to me, this is an analytical artifact.
I agree. This is all kind of, you're right. The signature is rich in like myofibroblastic cells, and that's clearly, a stromal contamination. So actually, maybe it's another way, an opportunity to kind of take a deeper dive into that subtype if there is room to investigate it. So another question: are CAP39L and CAP39L controlling the expression cells adhesion molecules? Is this part of their function, like normal function, perhaps?
Okay. I do not believe that they control adhesion molecules. They control those pathways which are involved in urothelial. They are upstream genes above PPAR gamma and CAP39 and TD62. So these are, I do not, each of these genes is, when you knock it down, of almost 1,000 genes. So these are massive phenotypic effects if we manipulate those genes. I do not remember whether the artificial molecules were coming on the top of this differentiation.
And just to follow up on my end, do you know like any function for these genes like in other organs? Is there like, do we know anything about them and how of their potential role in other organs, Dr. Shani?
The LPAR6 is a G protein. It's in the G protein pathway, and CAP39L is a calcium channel. And both
Sorry, we didn't hear that. I think your voice is breaking up. Yeah.
LPAR6 is a G protein encoding gene, and CAP39L is actually the calcium channel. Both of them are membrane proteins.
Okay.
They are, we did extensive study published many years ago of downregulation and methylation pattern of in multiple cell lines of different organs. And they are massively downregulated in many organ sites. So there is a potential that these genes may play a regulatory role or contributory role to carcinogenesis developing in other sites than bladder.
Interesting. Yeah, makes sense.
So,
yeah, Rohit.
Well, if that's okay, I think we have time for just one more question. Perhaps I can ask the last question and we can wind up. Is that okay, Hikmat?
Yeah, sure. Go ahead.
Okay. So last question for today, Dr. Cherniak, is sorry, we have more questions, but we'll try to address them offline. And one question we have is bladder cancer is more common in males than females, and do you feel male urothelium is perhaps more susceptible to field effects?
In other words, there is a mystery of sex or gender differences in bladder cancer. The common historical concept was based on occupation. Males were more often smokers. They were involved in occupational exposures, okay? And that was believed the source of this male. For example, orange agent related bladder cancer in Vietnam veterans is almost exclusively male. Okay? So it was the conventional approach was that this was not gender related, but this was related to the occupations or habits which were more common in males than in females. However, this approach is not entirely correct because the animal experiments indicate that actually, to simplify the story, that bladder cancer is much more difficult to develop in female mice than gender-related predisposition to bladder carcinogenesis. And I don't have a evidence whether the field effects of LPAR6 or CAP39 are much more in males or in females. But based on the analysis of TCGA cohort, I don't see that major difference in the methylation patterns of males versus females in bladder cancer.
Thank you, Dr. Cherniak. Hikmat, it seems we have just two more quick questions. Would you like to please like to have them addressed by Dr. Cherniak?
Yeah, I mean that's fine. If that's fine, since we still got a good number of attendees, so maybe I can ask the question on. Thank you for your very useful insights on pathogenesis in bladder cancer. What is your experience in any of these signature mutations translating in protein expression and being detected by immunohistochemistry in FFPE tissue?
The answer is very quick, 0.
Okay.
In other words, the mutations in bladder cancer are the most frequently mutated gene is P53. And we have an immunohistochemical test which is associated with the wild type P53 expression and contact. So this is a signature approach addressing this kind of issue. But they were proven to be ineffective in terms of the detection of bladder cancer or classification of perineoplastic intrauterine conditions. For example, I've seen major errors based on classification of small urothelial biopsy when you use the immunohistochemical staining for p53, and because it is positive, we call it carcinoma in situ.
I think we have one last question, maybe if we can. Dr. Shunya, great talk. I wonder if LARP6 and CAP39L are involved in your endocrine differentiation of urothelial carcinoma or not.
Can you repeat the last
So are the genes LPAR6 and CAP39 involved in your endocrine differentiation?
No, okay. So I do not believe that this is the case, okay? But basal tumors have a tendency that if you look at especially cell lines, they upregulate neuroendocrine markers. Okay? There's this kind of neural switch, okay? And this is most relevant when we develop to mesenchymal transitions. And of course, it's a signature of small cell carcinoma. But it is also upregulated in a good number of dominant basal organs. Whether this is on the whether these genes are direct targets of LPAR6 or CAP39, I don't have evidence for that.
Okay. Yes, that's all the questions. Thank you. Thank you very much. And I appreciate all your time and your you know, and you sharing your experience with us. Rohit, you have the final word.
Yeah, thank you, Hikmat. Again, before we close, like Hikmat mentioned, I also want to express my sincere appreciation to our speaker, Dr. Cherniak, for his time, expertise, amazing presentation, and thoughtful contributions to today's discussion. I think we got some great questions and an engaging audience, which was, I think, great. I'm thankful to our session moderator, Dr. Al Hamidi, and thank you everyone again for joining us today. We look forward to continuing these important conversations in future GUPS events. I hope all of you have a good day. Thank you and bye.
Thank you.